Okay, the start of a new chapter. This chapter consists of 3 modules also, and this chapter is dealing with neuroscience and behavior. And so it's going to get very science-based, very technical, very biological, and we're going to talk about the brain and how your brain actually affects behavior, which is one of the main reasons why we cover this chapter in psychology. Most students, want to help people and deal with disorders and therapy, and they don't understand why we have to discuss the brain and science in psychology. And I always explain to them that how can you actually treat someone if you don't know how the brain works? Because the brain and behavior go hand in hand. You may become a therapist, and let's say that you're going to treat someone with bipolar disorder. How are you going to know which medication to prescribe unless you know how that medication affects the brain, and the brain will then affect their behavior? So this chapter is extremely important in the study of psychology. So with module 7, the first module we're going to talk about neurons and how neurons are the basic elements that deal with behavior. And we have 4 questions we are going to answer in this module. The first one is, why do psychologists study the brain and the nervous system? The second question, what are the basic elements of the nervous system? Third question, how does the nervous system communicate electrical and chemical messages from one part to another? And then the fourth and final question of this module is behavior neuroscience, or the last thing we're going to cover, I shouldn't necessarily say a question, is behavior neuroscientists or biopsychologists. We're going to talk about how those are the psychologists who specialize in considering the ways in which the biological structures and functions of the body affect behavior. So there is a huge section, as I mentioned, in psychology that deals with neuroscience and biopsychology. And this is all about studying your brain and how your brain affects behavior. All right, so let's start out talking about neurons, right? And the structure of neurons themselves. Neurons are basically nerve cells, and they are the components of the nervous system. You have the central nervous system and you have the peripheral nervous system, right? The central nervous system consists of the brain and spinal cord, and the peripheral nervous system consists of all the other cells throughout, or all the other parts throughout your body besides the brain and the spinal cord. So it is those nerve cells which are the basic components of the nervous system. Each neuron within your body has a cell body, and in each cell body is a nucleus. It is the nucleus of each cell that contains your DNA or your hereditary information. So when you watch those crime shows and they're extracting DNA to see who committed the crime or who the baby daddy is, right? They're going into the nucleus of a cell body of a neuron in order to extract that information. So you have billions of cells throughout your body. Each cell has a cell body. Each cell body has a nucleus, and that is where your DNA resides, in that nucleus. Now, neurons themselves, there are two different cells in your body. There are neurons and there are glial cells, right? We just talked about how neurons communicate information from one part of your body to the next. The neurons are physically held in place by glial cells, which are the other type of cell in your body besides neurons. And glial cells do 4 different things within your bodies besides just holding the neurons in place. They provide nourishment to the neurons. They insulate the neurons. They help in repairing damage that is done to the neurons. And then the fourth and final function of the glial cells is that they support neural functioning. They support neural functioning. So, I mean, sometimes I like to think of neurons as the main cells of your body. However, without the glial cells, there would be no neurons. Neurons do all of the communication between other cells in your body, but it's the glial cells that keep those neurons intact. So in contrast with most other cells, neurons can communicate with other cells in the body. A lot of your other cells, your glial cells, do not do that, but the neurons can communicate with other cells. They can transmit information across relatively long distances. And so if you think about if some sort of stimulus, let's say you're walking barefoot and you step on a nail, right? So there is that stimulus of touch that is hitting your foot. That information has got to go up to your brain so your brain can figure out the message and then travel back down to your foot so that you lift your foot. From your foot to the brain is a relatively long distance. And it is the neurons that's going to transmit that information up to your brain. Whenever I keep saying transmit this information, right? Whenever any stimulus hits one of your senses, right? Your eyes, your nose, your mouth, your ears, or your skin. Whenever any stimulus hits those senses, there is a message or an impulse that generally moves in one direction. So it may start at one end of the neuron and move to the other end of the neuron. So it generally moves in only one direction. So let's talk a little bit more in depth about the structure of the neuron. And I think this will make more sense what I'm talking about whenever I say that it moves in one direction. In just a couple slides, I have a picture of a neuron. And so we'll get to physically see these different parts or these different structures of the neuron. At the very end of one neuron are what are called dendrites. Dendrites look like branches of a tree. They extend out from the cell body and they pick up incoming information from other neurons. Once they pick up this other information from other neurons, they send that information onto the cell body. From the cell body, the information goes to the axon. The axon is this elongated tube of the neuron, and it carries the messages that are received by the dendrites to other neurons. The axon is a very important part of the neuron because that is where the action potentials take place. And we'll talk more about that in just a little bit. So information comes into the dendrites, goes to the cell body. From the cell body, it goes to the axon. Once that information travels down the axon, it reaches the opposite end of the neuron, which is where you have what are called terminal buttons. This looks like a small button or bulge at the end. And so that information that has now traveled down the axon reaches the terminal buttons, and then it is released into what is called a synapse. And we'll talk about that in just a little bit too. Now, I want to go back to the structure of the neuron, and I want to go back specifically to the axon. The axon is covered and insulated and protected by what is called a myelin sheath. It is this fatty substance that wraps around and protects the axon. The myelin sheath is a fatty substance that wraps around and protects the axon. Now, there are certain places in the myelin where there are breaks. Those breaks in the myelin are called the nodes of Ranvier. Where the nodes of Ranvier are, the axon is not protected. And so at those nodes of Ranvier, information can come into and out of the axon. So the myelin sheath can speed up or slow down information traveling along the axon. So it serves to increase the velocity with which electrical impulses travel through the axons. Because it surrounds and insulates the axon, information cannot get into and out of the axon, except for where those nodes of Ranvier are. So the axons that carry the most important and urgent information, those axons have the greatest concentration of myelin. There aren't going to be as many nodes of Ranvier on these axons that carry the most important and urgent information, because you want the information to travel faster. Sometimes it could be a life or death situation, and so that information may have to go from your foot, from neuron to neuron to neuron to neuron, to your spinal cord, to your brain. Your brain figures out what the message is. It then travels again by neuron to neuron to neuron to neuron down to your foot, and you lift your foot. But you can see, depending on what you step on or what is occurring, it could have to travel faster. And so the more myelin you have, the faster the information travels. This is figure 1, and this is found on page 55 in your textbook. And this is a picture of a neuron. And so you can see the parts that we just described, right? The structures. Here are the dendrites. See, they look like branches of a tree. The dendrites pick up the information from their neurons, sends that information to the cell body. From there, it goes to the axon. These blue parts are the myelin sheath, and you can see how it wraps around and protects the axon. But you can see here are those breaks, right? Those are the nodes of Ranvier, where the axon is not protected. Once information travels down the axon, it reaches the terminal buttons, and from there, it will be released into the synapse. And then there's another neuron over here, and information gets sent from neuron to neuron to neuron. Now, if you remember what I just said on the previous slide, I think it's where I said that information generally moves in one direction, right? Generally moves in one direction. So information generally gets picked up by the dendrites to the cell body, to the axon, to the terminal buttons. It usually does not go backwards. So that's what I meant whenever I said that the information in a neuron usually travels in one direction, from the dendrites to the terminal buttons. Now, in order for information to be sent from neuron to neuron to neuron, there's a lot that has to occur in one neuron in order for that information to be sent onto the next neuron. And so the neuron actually has to fire. So in the next couple of slides, we're going to talk about how the neurons actually fire the information. Neurons follow what is called an all or none law. They either fire or they don't. When they do fire, that information will continue down the length of the axon. It will not stop. So if an action or if a neuron fires, then the process is in motion, or it will not fire. Either way, right? It's an all or none law. There's none where it starts and then stops. Now, let's talk about the axon itself. When a neuron is not firing, we say that the axon is in a resting state. It's the state before the neuron is triggered, and so the resting state of an axon or of a neuron is about -70 millivolts. And that's the electrical charge inside the axon, inside the neuron. When a neuron is at rest, the electrical charge inside the axon is about -70 millivolts. Now, when the axon or when the neuron does fire, we call that an action potential. This is where you have an electric nerve impulse that travels through a neuron's axon when it is set off by a trigger or this charge, this firing that occurs. And it changes the neuron's charge from negative to now positive. Remember, we said at a resting state, the inside of the axon, the inside of the neuron, is about -70 millivolts. However, when the neuron fires, the inside of that neuron will now become more positive as opposed to negative. That is when an action potential occurs. This slide is figure 2, and it is on page 57 in your textbook. And you can see what we're talking about with the firing that happens within a neuron, right? So if we read at the bottom, we have this positive charge, the negative charge, and the direction of the impulse, right? So it says the movement of an action potential along an axon. Just before time 1, positively charged ions enter the cell membrane, changing the charge from negative to positive, and therefore triggering an action potential. The action potential travels along the axon as it is illustrated in changes occurring from time 1 to time 3. So you can see here's the action potential, right? Goes from negative to positive, then it goes to time 2, the action potential goes from negative to positive, and then to time 3, action potential goes from negative to positive. This is the whole way down the length of this axon. There are many action potentials that are occurring. Now, as soon as that action potential is passed through a section, those positive ions are pumped back out, and the negative ions come back in, returning it to its resting state. So as soon as it leaves time 1, right, and goes to time 2, here back at time 1, that is returned back to negative again. And now it's more positive at time 2. Once that impulse travels from time 2 to time 3, time 2 now goes back to negative, and time 3 becomes more positive. So it does not stay in this heightened positive state. It has to return to its normal negative charge. Figure 3, again, on the same page, on page 57 in your textbook, this gives you an even closer up version. You can see the inside of the axon. We said in its resting state was more negative, right? The outside is more positive. You can see the negative ions inside and these positive ions outside. When an action potential occurs, these positive ions come in and the negative ions go out. But once that action potential passes, the positive goes out and the negative comes back in. Now, if you remember, we said that the axon was covered in a myelin sheath, except where there were the nodes of Ranvier. The nodes of Ranvier are the only places where these positive and negative ions can cross the channel. Only at the nodes of Ranvier. They cannot pass through the myelin. So you can see the resting potential right here at this graph on the right-hand side, negative 70 millivolts, right? And you have this burst, this action potential. It gives this brief reversal of charge within the neuron, and then it comes back down. But you see how it kind of overshoots? It goes a little bit further than negative 70. And then it comes back up again to its resting state. So you have the resting state, negative 70 millivolts. You have this burst of energy, right, of potential, where you're going up to about positive 40, and then you're coming back down after it has passed. But you overshoot and go even lower than negative 70. And then it slowly works its way back once again to that negative 70 millivolts when an action potential occurs. Now, if you remember, I said that an axon that is covered in myelin, the more myelin that there is, the faster the speed of transmission, right? Because there aren't as many places for information to go into and out of the axon. So I want to talk a little bit about this speed of transmission along the axon. So the speed at which an action potential travels along an axon is determined by various things, two different things specifically. The speed at which an action potential travels along an axon is determined by 1, the axon size, and 2, the thickness of the myelin sheath. The speed at which an action potential travels along an axon is determined by the axon size and the thickness of the myelin sheath. The neurons themselves can differ in various ways. So the quickness of an impulse moving along the axon, that is one way that neurons can differ. They can differ in their quickness of an impulse moving along the axon. They can also differ in terms of their potential rate of firing. And it is the intensity of the stimulus that determines how much of a neuron's potential firing rate is reached. The more intense the stimulus, the more the firing rate is going to be reached. I want to spend just a minute or two, or a slide or two, I should say. I don't know if it'll be a minute, but a slide or two talking about mirror neurons. I think mirror neurons are actually so cool. A mirror neuron, whenever you look in a mirror, right, you see something, you see yourself. Well, what happens with a mirror neuron, right, is if I'm looking at someone and I see them smile, I myself may smile. I may not even be aware of it. But that is what mirror neurons do. They mirror what else is occurring. I remember years ago, my niece and I were at a summer festival. It's actually called a corn festival, right? And I was feeding her this cob of corn. She was still quite little. And someone had taken a picture, and that picture had gone into the local newspaper. Here I am feeding my niece this cob of corn. And as I'm moving the cob of corn to my niece's mouth, of course, her mouth is opening because she's going to start eating the corn. What I noticed is really interesting in that photo in the newspaper is my mouth is also open. I was not eating the cob of corn. I was giving it to her to eat some corn, but my mouth is also open because that is mirror neurons in place. And I thought it was such an awesome example of a mirror neuron. So mirror neurons themselves, they are specialized neurons that fire not only when a person enacts a particular behavior, but also when the person simply observes another individual carrying out that same behavior. So my neurons are going to respond one way if I am smiling and happy, or if I am crying and sad. But if I see someone else smiling and happy, or if I see someone else crying and sad, my neurons are going to respond the same way as though I was the one who was doing that. All right, so let's talk a little bit more about mirror neurons. The discovery of mirror neurons suggests that humans' capacity to imitate others may be an inborn behavior. It may be innate, the way that we can imitate others, right? Because if you smile and then I smile, but I'm not even realizing that I'm smiling, that's some sort of reflexive reaction, this innate thing that occurs with these mirror neurons. It also helps explain how and why humans have the capacity to understand others' intentions. Mirror neurons are the basis for our feelings of empathy, right? If I see you really sad and upset, and then I myself feel sad and upset because I've been in the same situation, that is where I feel empathy. I'm empathizing with you because I know what you are going through. It's also the possible basis for a development of language in humans, because we are going to mirror what we see other people doing, what we hear other people saying. If we locate these mirror neurons, we may be able to stimulate them. And in stimulating these mirror neurons, that could actually help stroke victims. For many stroke victims, there is this paralysis on one side of the body. Well, because the mirror neurons are in place, if you can stimulate them, that may help with some of that paralysis. It may also help individuals with emotional problems. Now, I want to talk next about where neurons meet. This is this bridging of the gap, and we're going to talk about this quite a bit in the next couple of slides. If you remember, I said that information gets picked up by the dendrites, it moves on to the cell body. From the cell body, it moves down the axon to the terminal buttons, gets released into the synapse, and then gets picked up by the dendrites of a neighboring neuron. And that's how information goes from neuron to neuron to neuron until it reaches your brain. So I want to talk about where these neurons meet. So at the end of an axon are terminal buttons. That information is released into what is called a synapse. The synapse is the space between two neurons. It's where the axon of a sending neuron communicates with the dendrites of a receiving neuron. And it does so by using a chemical message. Now, when information gets picked up by the dendrites and moves on to the cell body and then moves on to the axon, and we were talking about it being electrical information, right? As that electrical information moves down the axon, it gets changed into a chemical information. From an electrical message to a chemical message. Those chemical messages are referred to as neurotransmitters. They are the chemicals that carry messages across the synapse to the dendrite and sometimes even the cell body of a receiving neuron. And we have a lot of different neurotransmitters. We have dopamine, we have serotonin, we have norepinephrine. We have many different types of neurotransmitters. Each neurotransmitter is very important for various behaviors. That's why neurotransmitters are so important in the study of psychology, because psychology is the study of behavior. So information starts out as electrical messages. It then gets changed into a chemical message, and it is the neurotransmitter that is that chemical message. It gets released into the synapse and then gets picked up by the dendrites of a neighboring neuron. This is figure 4A. I'm going to show you 4B in just a minute, but figure 4A on page 59 in your textbook. And you can see that there are 4 steps involved with this. So here are your neurotransmitters. They're reaching the terminal buttons, right? They are being released into the synapse. And you can see in step 3 now, you can see how these neurotransmitters are released into the synapse. These are the dendrites of a neighboring neuron. So these neurotransmitters are released into the synapse and they get picked up by the receiving end of another neuron. So it is those dendrites that get picked up. But one important thing to mention is that certain neurotransmitters need certain receptors. If dopamine is released into the synapse, then you have to have a dopamine receptor pick it up. You cannot have dopamine released and serotonin pick it up. That's not the way that it works. So it has to fit kind of like a lock and a key. There's a specific key shape to each lock. There is a specific neurotransmitter for each receptor. And so if you want an even closer look to this, that is on the next slide. So this is figure 4B, still on page 59 in your textbook, but this is even a closer look at it. Here are the information, right? The neurotransmitters, they're coming down the axon. They are here in the terminal buttons. They are being released into the synapse, and then they are being picked up by the dendrites of another neuron. So they are the receptors for those neurotransmitters. So let's let's continue on with bridging the gap, this place between where one neuron releases neurotransmitters into the synapse, and then the receiving neuron picks up those neurotransmitters and continues on the message. So not every neuron, though, is capable of receiving the chemical message carried by a particular neurotransmitter. If you remember, I said it needs to fit like a lock and a key. So if dopamine is released from one neuron and it crosses the synapse, it needs to be picked up by a dopamine receptor. If there is not a dopamine receptor, then the message does not get transmitted. So that's what this means by not every neuron is capable of receiving the chemical message. However, if there is a dopamine receptor to pick up that dopamine neurotransmitter, then the successful chemical communication is possible, and the information goes on to get passed. But it has to fit like a lock and a key. Dopamine needs to be picked up by a dopamine neurotransmitter. Serotonin needs to be picked up by a serotonin neurotransmitter, and so on and so forth. Let's talk about the types of chemical messages that are delivered by neurotransmitters. You can have both excitatory and inhibitory messages. So excitatory is a chemical message that makes it more likely that a receiving neuron will fire and an action potential will travel down its axon. The inhibitory is the opposite. It's a chemical message that prevents or decreases the likelihood that a receiving neuron will fire. So if there is again, a lock and key, the neurotransmitter gets picked up. If there is that correct receiving neuron, then that's an excitatory message. If it's an inhibitory message, then it decreases the likelihood of receiving the neuron. That will make it more likely for the information to get passed. Now, this flow chart, kind of shows you how this all works. So in the first block up here in the upper left-hand corner, it says, if neurotransmitters remained at the site of the synapse, it would produce constant stimulation or constant inhibition of the receiving neurons. And if that happens, then effective communication would no longer be possible. And so in order to do this, it's not as though those neurotransmitters are just floating endlessly out in the synapse. They either need to be deactivated or they need reuptake to occur, where they get reuptook, I guess that's a word, up into the the one neurotransmitter, and the process starts again. So it says, to avoid this problem, enzymes deactivate the neurotransmitters, or more commonly, the terminal button sucks them back up. And this is what reuptake is. It's reabsorption of the neurotransmitter by a terminal button. So again, one neuron releases the neurotransmitters from the terminal button into the synapse. They go into the synapse. There has to be the correct receiving or receptor neuron to pick up that neurotransmitter. If not, then two things can occur. Either an enzyme will deactivate that neurotransmitter, or reuptake will occur, where it's reabsorbed back into the terminal button and released again. And hopefully, then the second time it's released, there is the correct receptor of that receiving neuron to pick up that neurotransmitter. So you can see this process that occurs once the neurotransmitter is released into the synapse. Now let's talk a little bit more about this reuptake that could occur. The current understanding of the reuptake process has led to the development of certain drugs that treat psychological disorders. Because remember, this is psychology, right? And so if you take a drug, let's say for bipolar disorder, that drug is going to activate a neurotransmitter. So there are certain drugs that lead to the development of the treatment for that psychological disorder whenever reuptake occurs. So some of these are SSRIs, and that stands for selective serotonin reuptake inhibitor. And what these SSRIs do is they permit certain neurotransmitters to remain active for a longer period at certain synapses, reducing the symptoms of depression. So a lot of individuals who are diagnosed with depression, they will take a drug that is an SSRI. So it's a selective serotonin reuptake inhibitor. So it inhibits the reuptake of serotonin. So that serotonin remains more active in the synapse in order to be picked up by a receiving neuron. So neurotransmitters, they're actually these multi-talented chemical couriers, right? Because they transmit information. So they're a courier. If you think about what a courier is, a courier delivers packages or delivers messages. That is what a neurotransmitter does. So they are particularly important in linking the information between the nervous system and behavior. They are also important for maintaining vital brain and body functions. If there is a deficiency or an excess of neurotransmitters, this can produce severe behavior disorders. So a lot of times when you have a certain behavior disorder, it all stems from the neurotransmitters. Perhaps you have too much dopamine, or maybe you don't have enough dopamine. Perhaps you have too much serotonin or not enough serotonin, right? And so on and so forth. But a lot of the psychological disorders that we have stem from either an excess or a deficiency of certain neurotransmitters. We currently know that there are more than 100 chemicals that have been found to act as neurotransmitters. And neuroscientists believe that there actually may be more than that. They just are not yet identified. Now, this table is taken from, I think it's, page 61. This is figure 5, and these are our major neurotransmitters. These are not the only neurotransmitters that we have, but these are the major ones. And you can see with this is a really nice table. It shows you like acetylcholine, glutamate, GABA, dopamine, serotonin, and endorphins. It tells you where they are located. It gives you the effect. Are they excitatory or are they inhibitory neurotransmitters? And then the function for them. So acetylcholine is important for muscle movement and cognitive functioning. Glutamate is important with memory. GABA, eating, aggression, sleeping. Dopamine, important for pleasure, right? We think about dopamine being our feel-good neurotransmitter. So it deals with pleasure and reward and attention. Serotonin is very much involved in depression, also with sleeping and eating and mood and pain. And in endorphins, pain suppression, right? So if you're ever in a lot of pain, your endorphins may kick in so you don't feel as much pain. So they're the pleasurable feelings, the appetites, the placebos. Okay, so that ends Module 7.